Thank you so much, Caitlin, for the lovely introduction and for inviting me here. It is a pleasure and an honor to be here, and I've had a fantastic day meeting with a lot of people and hearing about a lot of really cool science. My head is sort of buzzing right now. I'm trying to remember what it is that I actually work on, but I'm thinking my slides will probably have some clues to remind me. So let's find out. So I want to start by telling you where I'm from. So I'm from Worcester Polytechnic Institute in the city of Worcester, second largest city in New England. We're about an hour west of Boston, and it's a fun little place to be. And benefits include, once you learn how to say Worcester, then you can talk about Worcestershire sauce with a lot of confidence in your pronunciation. So, all right. So what I want to talk about today is mycobacteria and how they regulate expression of their genes and some of the sort of non-canonical aspects of this. And so we're interested in mycobacteria because they cause some bad diseases, including tuberculosis. Tuberculosis is still a huge cause of disease and death around the world, with over 8 million cases, over 1 million deaths each year. And a big part of the problem with TB is it's difficult to treat. People have to take multiple antibiotics for 4 months or more. And this is a big logistical challenge, especially in developing countries with less developed healthcare infrastructure. So these are pictures I took when I was in Peru now almost 20 years ago, which is hard to imagine. I was able to go and spend some time with clinicians and people doing TB research there, and that was what really got me interested in and engaged in this field, and I realized this was the thing that I needed to be studying. So when we think about this problem of the extremely long drug treatment, we have to ask ourselves, why is it so hard to kill these bacteria? So this is Mycobacterium tuberculosis, which I'll call MTB. And here we can see these little bacteria happily or semi-happily living inside a macrophage, which is the cell type that's supposed to be killing them. So obviously that's not working very well. And so when we're thinking about this problem of antibiotics not being very effective, there are multiple contributors to this. But one of them is the stress responses of the bacteria to the stresses they encounter even before they see antibiotics. So these bacteria get engulfed by macrophages. The macrophages are trying to kill them with low pH, nutrient restriction, reactive oxygen and nitrogen species. The bacteria are fighting back and often winning, but still sort of having to deal with these stress conditions. These infected cells get engulfed in these structures called granulomas. And in the center of the granuloma, right here, there's a hypoxic zone because these aren't vascularized. And then we have bacteria sort of floating around in this hypoxic region. And they're obligate aerobes. So they don't like hypoxia. They have to stop growing. So they're sort of facing this slew of different stressors that vary depending on where within this lesion the bacteria are. And they're going to respond to them with their stress responses. And more often than not, those stress responses make the bacteria less sensitive to antibiotics. So here's an example of this problem. Here are some of the most important antibiotics for treating TB. And this study looked at how much higher a concentration do you need to kill the same proportion of your culture if you have bacteria that are happily growing in log phase versus growth arrested due to hypoxia. And so there's the fold increase in antibiotic concentration that you need. So that's a lot of antibiotic. And when you look at the targets, this sort of makes sense. So these are things that rapidly growing bacteria really have a lot of use for. And if they're sitting there in this sort of hunkered down semi-dormant state, they're not going to need their ribosomes so much. They're not making new cell wall anyway, so they just don't care as much when you interfere with those processes. So we have this sort of situation where we have these different stressors, and then depending on which stressors and which combinations, they often lead to things like metabolic reprogramming, reduced metabolic activity, total cessation of growth. And then this leads to this tolerance, antibiotic tolerance, where the bacteria are not efficiently killed because they're not metabolizing very much, they're not growing very quickly. And then that can lead to an even bigger problem, which is genetic antibiotic resistance, because then you have these populations of bacteria that are sitting around allowing mutations to occur, and eventually resistance can happen. And so I'm really interested in what's happening here, sort of trying to understand what is the biology that allows these bacteria to detect these stressors and then get into these hunkered down states and then cause this whole slew of downstream consequences. And so that's sort of the really big picture motivation for a lot of my work. And so when we think about, well, what does an organism do to respond to stress? Of course, you're going to regulate expression of genes. And we can typically think of this happening as regulation of transcription itself. But then there's also mRNA degradation, and that's also a regulated process. And so in bacteria in particular, when they're growing quickly, they're making and degrading their RNA very quickly. And so they have a lot of opportunities to regulate this. And it's the combination of transcription rates and mRNA degradation rates that actually give you steady state abundance of whatever transcript you're looking at. So when I started learning about this, one of the things I learned pretty quickly was that we don't really know anything about how mRNA degradation is regulated. And so that caught my interest because I like things that we don't understand and that are really hard to understand, it turns out. And so when we think about how this type of regulation can play out, we can think about a couple of different possibilities. We could have transcript-specific regulation where a particular RNA is more or less stable, and that contributes to these differences in gene expression patterns. And then there's also global regulation. In particular, in response to energy stress, mRNA degradation is globally slowed down. And this seems to be an energy-saving mechanism. It's not energetically efficient to make and degrade each RNA every couple of minutes. And so both of these are things that certainly are happening in mycobacteria, and we still understand very little about how they're happening. And so I want to show you a little data supporting this idea of this global mRNA degradation regulation in response to energy stress. So this was a really important study from M. tuberculosis. And here, this is an experiment where transcription is stopped at time 0 with a drug, and then mRNA degradation occurs and mRNA levels decrease at a rate that's due to hopefully, however they were degrading their RNAs before we came in and made that perturbation. And so when the bacteria are growing happily in aerobic conditions, they have very rapid degradation of their RNA. But here, when they are growth arrested in hypoxia, the mRNA pool is way more stable. And we found the same thing happens in our favorite model organism, which is M. smegmatis. So here we're looking at a histogram of genes, and here we're looking at half-lives. And we can see that in log phase here in dark blue, a typical half-life is 2 or 3 minutes. And in hypoxia, they're much, much longer. So clearly we have this big broad slowing of mRNA degradation. And it's not because the RNases themselves are downregulated. The RNases are still there. So it seems their activity is what's being regulated. And so how's that happening? Well, it turns out to answer that question, we have to know what are the enzymes that are doing the process. And we've done a lot of work trying to define what those are and what they're doing. And one of the components of this process, these processes are what I want to talk about for most of my talk today. So we have RNases, are endonucleases that cut within a transcript, and we have exonucleases that sort of chop Pac-Man style from one end or the other of the RNA, one nucleotide at a time. They're helped out by helicases that unwind secondary structure. RNA binding proteins can protect transcripts or sort of feed them to the RNases. And we have discovered some new components of this machinery. We recently found a new iron sulfur cluster protein that doesn't exist outside of the actinobacteria. I'm not going to talk about that today for the sake of time. If anybody wants to talk about iron sulfur proteins later, let me know, because I'm super excited about that. And so the star of today's story is an enzyme called RPPH. And this is an enzyme that prepares the 5' ends of transcripts for degradation. So in bacteria, in all organisms, a transcript is made with a 5' triphosphate. And in bacteria, there's no canonical cap like eukaryotes have. But these 5' triphosphates can function like a cap in protecting mRNAs from being degraded. And this has been shown in E. coli and in Bacillus subtilis and a small number of other organisms as well. So we have our 5' triphosphate, an enzyme called RPPH. This is RNA pyrophosphohydrolase, is going to come along and cut off a pyrophosphate, leaving a monophosphate. And the monophosphates stimulate degradation by some of the important RNases. So this can be a rate-limiting step in degradation of many transcripts. And so in MTB, in Mycobacteria, there was no annotated RPPH. This is my former postdoc, Ilario, who did most of the work I'm going to talk about today. He is fantastic. He decided he was going to find the RPPH. And we thought there had to be one because we had done experiments looking at the chemistry of 5' ends of mRNAs, and we knew that there are lots of mRNAs floating around in the cell that have 5' monophosphates. And so they presumably had to be coming from this activity. But we didn't know what enzyme was doing it. So Ilario did a little reverse genetic screen where he knocked out 10 genes that encoded proteins with a domain that's common in RPPHs. And then we looked at how these knockouts, we looked at how they affected the 5' end chemistry of a particular transcript. And so here we are very lucky that DNA and RNA ligases require a 5' monophosphate to ligate something onto a 5' end. And this is what gives us the power to study these things. So here, if we have an mRNA and we try to ligate an adapter and there's a triphosphate, nothing's going to happen. If we have a monophosphate, we'll get our ligation to occur. And so we can take advantage of this to assay the 5' end chemistry of a transcript that we care about. So what we do is we take our RNA from the cells and we treat some of it with RPPH from E. coli, which luckily we can buy from NEB, and we leave part of it untreated. And then we attempt to ligate on an adapter, and we can use quantitative PCR to see how much adapter was ligated. And so here we have a transcript that did not get ligated unless we treated it with RPPH from E. coli. So we know that it had a triphosphate on it at the point when we got it out of the cell. So we chose a transcript that we knew had a high degree of 5' monophosphorylation in the wild type strain. So we thought it had to be a substrate for this RPPH that we expected to exist. And we did this splinted ligation assay, and I'm not going to explain the y-axis here, except that higher on the axis means more triphosphates. And so here's our wild type strain, and here are these 10 different knockouts, and we had this one knockout with a huge difference. And this is one of those really beautiful results that you see, and it makes you so happy that you're doing science. So much of the time you do not see a result that is this beautiful and this clear. But every now and then it happens, and you feel really good. So this was great, except this gene was annotated MUT4, and it was annotated as being a nucleotide pool cleanser. These are enzymes that degrade nucleotides that are damaged by oxidative stress or other things that damage nucleotides to prevent those damaged nucleotides from getting put into the DNA. So MUT4 was annotated, mut is for mutator, as being part of this process in Mycobacteria, and there were a couple of papers that had data that was sort of consistent with that idea. So we were initially a little bit skeptical that this was really our RPPH, and we had to do a lot of experiments to convince ourselves and our friends in the world. So here we're looking at results from that same assay, where here's our wild type strain, and here's our MUT4 knockout, and we have this big increase in triphosphates. And so we found we could complement it by adding MUT4 back. That's super important. And if we added back a point mutant that's predicted to abolish the catalytic activity, then it didn't work. So it's due to the catalytic activity of this enzyme, not some other moonlighting function. And then we purified the protein. We overexpressed it in E. coli and purified it and gave it the same transcript in vitro. And now here we can see when we don't treat that transcript with any proteins, it has lots of 5' triphosphates. When we treat it with our MUT4, now we get lots of ligation. We have way less triphosphates. And that's dependent on the catalytic activity. So at this point, we were convinced, yes, this really is an RNA pyrophosphohydrolase. And so we're not going to call it MUT4 anymore. We're going to call it RPPH. All right. And so one of the pieces of data that had been used previously to conclude that this might be involved in oxidative stress response was that a MUT4 knockout in M. smegmatis was more sensitive to hydrogen peroxide. So we tested that, and we actually found in MTB, it's the exact opposite. So here we're looking at survival of an acute exposure to hydrogen peroxide. Here's our wild type strain, which hates this, and here's our MUT4 knockout, or RPPH knockout, which is now doing quite well. So this is exactly the opposite of what was previously found in M. smegmatis. And this is consistent with the idea that this enzyme's not actually doing anything to help with DNA damage response, but probably it's affecting expression of various genes, and those are affecting tolerance to oxidative stress and other things, and those effects may be different in MTB and smegmatis. That happens all the time. All right. And when we get a mutant, we want to look at its sensitivity to drugs and other stress conditions that we care about. And so we looked at sensitivity to drugs, and the strongest signal we found was for a drug called vancomycin, where we found that here our RPPH knockout is more sensitive to vancomycin. And so that's what we're seeing here. Here's our wild type strain in blue. And when we knock out RPPH in red, now its growth is inhibited at lower concentrations of vancomycin. And we can complement that phenotype. So we saw this for vancomycin. We did not see this for most drugs. And this led us to wonder if the vancomycin sensitivity had something to do with permeability of the cell envelope. So mycobacteria have really weird cell envelopes. They're sort of neither gram-positive nor gram-negative. They have peptidoglycan, and then they have some other layers outside of that, including this sort of non-canonical outer membrane. So this thing functions kind of like an outer membrane. It's a permeability barrier, but its composition, its chemical composition is totally different from that of the outer membrane of gram-negative bacteria. Vancomycin acts here. So it blocks peptidoglycan biosynthesis. It doesn't actually have to get into the cell. It just has to get into the periplasm here. So we wondered if this outer membrane might be more permeable in our RPPH knockout. We had no idea how to test that. So we found a collaborator who did. This is Irene Lepore in Sloan Segrist's lab at UMass Amherst. And she is an expert at looking at the permeability of the mycobacterial outer membrane. And so she did a couple of assays to look at that for us. And we see that indeed the MUT4 knockout, the RPPH knockout is more permeable. So here, this increased uptake of fluorescent vancomycin is telling us that yes, more vancomycin is getting through that outer membrane. And then this is another assay she did that I'm not going to explain because it's very complicated, but a lower value here means greater permeability. So now we have an idea of why this strain is more sensitive to vancomycin. But what's happening? Why would deletion of RPPH affect that? Well, we figured it probably had something to do with regulation of gene expression. And so we wanted to see if RPPH is acting more broadly. So far we'd only looked at its activity on one single transcript that was sort of arbitrarily chosen. And so to assess if it acts transcriptome-wide, we used a method that is sort of related to the ligation assay I showed you before. So here's our pool of RNA that we can get from a cell. And in this RNA, we're going to have different kinds of 5' ends. There'll be 5' ends that are derived from transcription initiation, so primary 5' ends, and they may have a triphosphate or a monophosphate, depending on whether they've been acted upon by a native RPPH. We also have RNases that are going to cleave and leave 5' monophosphates, and these are not important for the process we're looking at, but it turns out to be a really useful control that I'll show you in a minute. So we take this pool of RNA from cells, and we either treat it with E. coli RPPH or leave it untreated, and then we ligate on an adapter. And similarly to what I showed you before, we're going to get some transcripts that are only going to get ligated or get ligated at higher levels if we treat them with RPPH to remove the triphosphates. And so then we can look at the ratio of reads we get for a particular 5' end in this RPPH treated library and this untreated library. And that gives us a metric of how much triphosphates were there in the first place to get removed. And so here we're looking at our wild type strain, and this log2 coverage ratio of the higher number here means more triphosphates. And here's where our cleavage sites come in handy. So we knew that the cleavage sites all have monophosphates to begin with. So their ratio should be right at 0 on this log2 scale. So that's our internal control to let us know we did the normalization right, which is actually a kind of tricky thing to do with this type of experiment. And then here are our transcription start sites with these higher ratios. And so now in our RPPH deletion mutant, we have this population of transcription start sites, but their log2 coverage ratios have shifted over. And this was another one of those beautiful results where we saw this and we knew, okay, yes, this thing is acting transcriptome wide. It's affecting the 5' ends of lots and lots of different transcripts. And thankfully, it complemented when we added back RPPH. So we concluded that yes, RPPH is acting transcriptome wide, but on some transcripts a lot more than others. So if we look at any particular, we have this bulk population here that's sort of moving over to the right of the axis, but some transcripts have big changes and some transcripts have small changes. So it's not acting equally on all of the 5' ends. It's going to affect some transcripts more than others. And so we looked at the characteristics of the transcripts that correlate to higher impact by RPPH. And we found that RPPH seems to prefer unstructured 5' ends, which was not super surprising because that had also been shown for RPPHs in E. coli and Bacillus. We found that there's a preference related to actually the nucleotides. So here, the higher the value, the greater the impact of RPPH on the 5' end. And we see that some 5' ends, like those starting with GA, are affected much more than others. So there's this strong nucleotide bias that turns out to be different from what we've seen in other organisms. And maybe that makes sense because these different organisms have different GC percentages in their genomes. Okay, so we have some idea of how RPPH is choosing its substrates. And now, according to the model, this should sensitize transcript to being degraded. And so we had to test that. So here we make our favorite transcript in vitro with its triphosphate. We treat it with RPPH or not. And then we give it purified RNases. So RNase J and RNase E are other RNases that have sort of defined roles in mRNA degradation in mycobacteria. And we did this with the help of Jim Saccatini's lab that did some of the RNase purifications for us. And so here we have our transcript, and we're just looking at how much gets cleaved by the RNases. And we see here when we pre-treat with the wild type RPPH, we see a big increase in cleavage by RNase E compared to if we don't pre-treat it or if we pre-treat it with this catalytically dead mutant. So that was totally consistent with our hypothesis. We looked at RNase J, another important RNase, and we see something similar, a smaller effect here, but still RPPH treatment helps sensitizes the transcript. So far so good. This is what we expected. And so then to put it all together, we wanted to show that the same thing happens in cells. It should also be sensitizing transcripts to degradation in cells. And so to look at this, we need to measure mRNA half-lives. And so we do this by treating with rifampicin, a drug that's going to block transcription. And then we can measure RNA abundance over time using quantitative PCR. And hopefully that rate of decrease in transcript abundance is reflecting the rate of mRNA degradation. So we get a curve like this, and we can calculate the half-life from looking at the slope here. This is decrease in mRNA abundance over time after we add RIP. Okay, and so our expectation was this. For any transcript we look at in the wild type strain, there'll be some degradation rate. And then when we knock out RPPH, that degradation rate should be less. The half-life should be longer because the transcript has its little 5' cap intact and it's less sensitive to these RNAs. And so now here are data. We have a funky y-axis here, but this is essentially a log2 measure of mRNA abundance. And our wild type strain is in blue, and our RPPH knockout is in red. And we can see here that indeed we have a slightly slower rate of degradation in the RPPH knockout. And that's exactly what we expected to see. We looked at other transcripts, and here's an example of one that did not match our expectation. So here, for this transcript in the RPPH knockout, it's not being degraded more slowly. If anything, it's being degraded a little bit more quickly. And that's the opposite of what we expected to see. So maybe not. So we looked at 10 transcripts in total by qPCR, and we found that for exactly one of them, we saw what we expected. So here, this is our star transcript. We knock out RPPH, and we have a higher half-life. So here, now we're looking at half-life on the y-axis. For all of the other transcripts, we saw sort of the opposite. There was a trend towards faster degradation in the RPPH knockout, and in most cases, slower degradation in our complemented strain here, where all we've done is add back RPPH with its native promoter. And so I did this experiment myself. This was the first lab experiment that I had done in years. Ilario had finished his postdoc and he had left, and the reviewers wanted this experiment done. And so I buckled up and I went into the lab and I proved that I still knew how to pipette. And it went pretty well. So for this kind of assay, I want to tell you, these are really small error bars. They might look big, but this is a hard assay to do. But when I got these data, then I wondered, did I mix up my strains? Did I mix up my knockout and complemented strain? Because if so, that would kind of explain all these results. So I actually did a little bit of PCR to check. And no, I didn't mess up the strains. So I guess that's good that I know what I'm doing, but made these data a lot harder to explain. So most mRNAs are not stabilized when we delete RPPH. Complementation of the deletion strain seems to stabilize most transcripts. So there's definitely more going on here than we thought. And there's things going on here that we don't understand. And so we still don't fully understand it. So unfortunately, I don't have an answer for you that fully explains this, but we have some ideas. And so I need to tell you something else about RPPH, which is that the mycobacterial RPPH has two intrinsically disordered regions. So here we've got our alpha fold prediction, and here we've got a disorder predictor, where here we have this high disorder score for the N-terminal and C-terminal regions. So these are IDRs. And this was a little bit unusual. So RPPHs from E. coli and B. subtilis and other organisms that we looked at don't have these IDRs. So this seems to be like a special little mycobacteria feature here, but it is conserved among the mycobacteria. And so one thing that IDRs are known for is inducing phase separation. So undergoing liquid-liquid phase separation to make condensates within bacterial cells that might be hubs where certain processes can happen. And so this idea had been explored a bit already with respect to, well, lots of different processes at this point, but of particular interest to us, it's been shown for several RNases that they have IDRs that can trigger formation of these phase-separated condensates. And so this was shown really rigorously and nicely by Jared Schrader's lab in Caulobacter crucentus for an enzyme called RNase E, which I mentioned before. And RNase E is really important. So it has a rate-limiting role in degradation of maybe 80% of transcripts in mycobacteria. And so we looked at this in mycobacteria as well. And we see that in mycobacteria, RNase E also forms these foci that we refer to as condensate-like bodies, because we've not done all the proper controls to call them condensates. But it forms these foci, and these are dynamic. And so we see this if we treat with rifampicin, which blocks transcription, and it's going to cause mRNA levels in the cell globally to drop. These foci sort of diffuse out. So that means they're not aggregates that are artifacts of our fluorescent protein tag or something like that. They're these dynamic bodies. And here's quantification of that. And this is the work of a talented grad student in my lab, Abby Rapako, who actually defended her PhD just a few weeks ago. Okay, so Ilario wondered, is RPPH also forming foci through one or both of its IDRs? And so for this, we had to turn to M. smegmatis. Everything else I showed you so far has been with MTB. But in order to do this microscopy, we have to take it out of biosafety containment. And it turns out that fixation disrupts a lot of condensates in cells. So we can't actually look at this in MTB, but we can look at it in M. smegmatis. And so here we are looking at dendra-tagged RPPH, and we see it does form these foci. And in many cases, these foci overlap with foci formed by RNases. So here we're looking at that. We have co-localization with RNase E, the percentage of RPPH foci that co-localize with RNase E or with RNase J, and these are significantly higher than you would expect to happen by chance. And so this is consistent with an idea of RPPH having a role in RNA degradation and maybe localizing with these RNases to form degradation hubs. Okay. And we also put the MTB RPPH in M. smegmatis and found it did the same thing. So that helped give us confidence that our non-pathogenic model was helping us to study the thing that we wanted to study. Okay, so Ilario wanted to know if the IDRs were responsible for this. He made some IDR deletion mutants, and that seemed to destabilize the protein. So instead, he took the IDRs and attached them to dendra to see if they were sufficient to trigger foci formation. And so here we have these constructs that he made to do these experiments. And we found out that, yes, the N-terminal IDR itself is sufficient to trigger formation of these foci. And so that's what we're seeing here. We have just this N-terminal IDR fused to dendra, and we get our nice bright spot. So that doesn't happen. Dendra itself is diffuse as it should be, and the C-terminal IDR doesn't have that effect. All right, so we found this, this region of the protein that seems to be causing these globs of protein. And so now we can start to speculate about what might be happening. And this is the part where we do not have data to back this up. But we could speculate that maybe we have degradation hubs where we have RPPH and RNases together, and RPPH is decapping the transcripts and the RNases are right there and they're degrading it. And so I showed you that co-localization, but we also have RPPH foci that don't localize with RNases. So maybe we also have places where RPPH is binding to RNA and maybe acting upon its 5' ends, but just kind of hanging on to it and actually protecting it from degradation. And so that could potentially explain the sort of paradoxical result that we have of this in vitro activity not reflecting what actually happens in vivo. And so I want to tell you a little bit more about, give you a little bit of a broader picture of what we've learned about how the mRNA degradation machinery is localized within mycobacterial cells. So while Ilario was working on that project, my grad student, Abby, was also working on trying to understand the mechanisms and significance of subcellular localization of RNases. And so here, Abby's looking at correlations and localization of RNase E, that really important RNase, and 3 other components of the RNA degradation machinery. And so if we just look at these, the things that are boxed in red here, and here we're looking at Pearson's R values, so we're looking at correlation of the signal intensity for 2 different fluorescently labeled proteins. And we have these pretty high values for all 3 of these enzymes that we looked at. So we have these blobs within cells. We could call them foci or condensate-like bodies, or whatever it is the reviewers will be happy with us using, because they have strong feelings about this. Okay, so we have these localization patterns where we have these foci. And then if we look at cells that have been starved for carbon, which we use as kind of a tractable model for energy stress. Hypoxia is a little bit challenging to do if you want to do microscopy. And here we look at, now we're looking at foci per micron. So how many of these foci we see in cells corrected for their weirdly variable lengths. And we see that for most of these, so for 3 out of 4, we see a decrease in the number of foci when we move from log phase growth to carbon starvation. So these foci seem to be falling apart in carbon starvation. And we know that in carbon starvation, the mRNA pool is globally stabilized. So here we're looking using qPCR to look at a few genes. And in each case, we see that the half-life here in orange is extended in carbon starvation compared to in log phase growth. And so based on these observations, we like to speculate that these RNase foci could be sites of active mRNA degradation and that they dissociate in carbon starvation, and that this is part of the mechanism of what's stabilizing the mRNA pool. Now we have less efficient degradation because the pieces of the degradation machinery are all spread out throughout the cell and they can't just hand off the RNA from one enzyme to another quite so easily. So it's a nice model, right? And if you're anything like reviewer 2, you were wondering right now, how are we going to test this exciting model? And so I have to tell you, we have not figured out a way to test this that has been effective so far. So what we would want to do is say, okay, in log phase, we have these foci, these blobs of proteins, and can we artificially disrupt those and then see if mRNA degradation slows down, right? And so we've done some experiments to try to do that, and they've not been able to give us the data we want, but we've learned some really interesting things in the process. So I'm going to tell you just a little bit about that. So I said that RNase E itself has IDRs that it uses to form these condensate-like bodies within mycobacterial cells. This is the AlphaFold prediction for RNase E, and you can see it looks like a hairball. This is a horrible protein to work with, as my poor grad student learned. But luckily she persevered, and she and another grad student made some deletions, so we can delete one or both IDRs and test what the impact of that is. And together with Vidya in Jared Schrader's lab, we were able to look at how this impacted formation of these condensates in vitro. So these proteins will form these blobs in vitro if they're present at high enough concentrations. And when we delete both IDRs, we lose formation of these condensates in vitro, suggesting that the IDRs are necessary for condensate formation, which is not a surprise. We expected that because that's what was already shown in Caulobacter crescentus. The IDRs also have another function, which is interacting with other degradation proteins. And so this was totally, these interactions were completely unknown when Abby started this project. She heroically did many, many reciprocal co-IPs and was able to identify direct interactions between 3 other components and determine that they all involve the IDRs. So we can delete the IDRs and also abrogate those interactions with other parts of the degradation machinery. Okay, so we can make our IDR-less RNase E that doesn't make blobs in vitro. It still makes foci in cells. So here we're looking at foci per micron. This is full-length RNase E. And here in purple is our IDR-less deletion mutant. And it has maybe a small decrease, but not super dramatic, not even statistically significant in this figure. So although it uses those IDRs or needs those IDRs to make these condensates in vitro, something different is happening in cells. We also can look at the co-localization of RNase E with its binding partners, which we expected might be affected if we remove the domains or the regions of RNase E that it uses to undergo those interactions. And so here, there's a lot going on this slide. But if you just compare the purple to the green, purple is where we've deleted both of the IDRs, and green is the full-length protein. And so in one case, this had the expected effect of decreasing the correlation, the localization of the 2 proteins. And the other case, the other 2, it had no effect. So what we sort of take home from all this is that the subcellular localization of these proteins within mycobacterial cells is really robust. There seem to be multiple different things that are causing these things to localize in the places where they do. And because of this, it's really hard to break the system. So we thought we would have broken it in two different ways here. And instead, we sort of didn't break it in any ways at all, which is sort of frustrating for us. I think it's interesting to think about in terms of how we sort of integrate our understanding of what we see in vitro and what we see in cells. Because here now we have sort of two examples, one with RPPH and its impact on degradation by RNases, where we kind of see opposite things in vitro and in cells. And then here we see with RNA-seq and its localization patterns, that things that disrupt what we think are the determinants of that localization in vitro then don't have the expected effect in cells. So I guess it speaks to the importance of looking at things acting within their native environments, which is sort of unfortunate because trying to do cell biology in bacteria is not fun. I don't recommend it. Okay, so rolling back to RPPH here now, I can summarize what we found. So we sort of defined these molecular functions for RPPH. It's definitely removing pyrophosphates, and it's sensitizing transcripts to RNases in vitro, although in vivo seems to be a different story. It forms these condensate-like bodies with or without RNases, and it affects sensitivity to environmental stressors in different ways, increasing sensitivity to vancomycin by increasing the mycomembrane permeability, but then deleting RPPH makes these cells more resistant or more tolerant to hydrogen peroxide. And so we have lots of outstanding questions. Of course, we want to know how this sort of paradoxical stabilization effect happens. So we're working on that. We want to know what is the mechanism by which our RNA 5' end status affects phenotypes like this, the outer membrane permeability and the sensitivity to oxidative stress. And we expected that might be through regulation of gene expression. I think I told you that earlier, and then I didn't tell you about any genes that explained it, and that's because we haven't found those genes yet. There's actually less differences in steady state transcript abundance in the RPPH knockout than we would have expected. And that has led us to wonder if the impact of this 5' end modification might be in part related to translation as well as to transcript stability. And so we want to directly ask that question. And this is sort of a plausible idea in mycobacteria because a lot of mycobacterial transcripts are leaderless. They have no 5' UTR. So the very 5' end of the transcript has to somehow feed itself into a ribosome for translation to begin. And you can imagine that the chemistry of that 5' end could plausibly affect that process. So we'll see. We've not yet tested to see if RPPH directly interacts with other RNases. And in part, this is because it is even worse behaved than RNase-E. It sort of phase separates in vitro if you look at it wrong. So it's been a little bit hard to work with. And we'd like to know what else is present in these RPPH containing bodies, because that might also give us clues as to sort of what the physiological impact of this, this protein is. All right, so I'm going to stop there. I want to thank the people in my lab who did the work, Junpei, who did the bioinformatics, Abby, who did a lot of the RNase-E work, Ilario, who has sadly moved on from us and did most of what I told you about, our fantastic collaborators, especially Sloan's lab, and we have funding from NSF, NIH, WPI, and the POTS Foundation. And thanks to all of you, and I'm happy to hear your questions. I would love to know what's different about that one transcript. I had a lot of good ideas and they were all wrong. So I thought maybe it has to do with the first nucleotide, whether it's an A or it's a G. Maybe it has to do with whether it's leadered or leaderless. And I've not found anything yet that can explain that. That would be great to know. I have no idea. So one thing that you will notice we have not looked at in any of this is where the RNAs actually are. And the reason we haven't done that is because it's really hard to do. But it is theoretically possible, and I think that would be a really interesting question to ask, because if we think about where the transcripts are localized, that might give us answers to some of these outstanding questions. Yeah. 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 How many Nudix hydrolases are there? In MTB, there are 11. There are the 10 that Ilario knocked out, and then one that was essential. Yeah. Oh, yeah, yeah, like non-canonical initiating nucleotides. Yeah. We don't know that yet. There is a NUD-C, which is an enzyme that we would expect specifically to remove NAD or NADH caps. And so that's something that we want to look at, but we haven't looked at whether RPPH has activity on them. Yeah. Yes, yes. Yeah. Yeah. Yeah, okay, the AP4A, I actually have not thought about that in a long time, and I should think about that more. Can take it off. Yeah. Yeah. That would be really interesting to look at. Yeah. Yeah. I can't remember the order, but yeah, so I showed you the pyrophosphate getting chopped off in one fell swoop, and apparently in E. coli, that's not the case. Yeah. Yeah, so we sort of considered that idea. I guess the thing that we know is we know that RPPH gives you a monophosphate because it makes the 5' end ligatable. So we know that it can remove the pyrophosphate, maybe one nucleotide or one phosphate at a time, or maybe all at once. And we know it must be doing that in cells as well, because when we pull the RNA out of the cells, we have more 5' monophosphate of that other thing. Not that I specifically know about. I don't remember if that one is also a Nudix hydrolase or not. Yeah. But we couldn't really tell. It was hard to tell from looking at the Nudix hydrolases which one was going to be the RPPH, which was why we just knocked them all out. Yeah. It is similar. So it has a Nudix domain, which is there's amino acid sequence in common, and then its predicted 3D folding is also related to the E. coli RPPH. The preference for unstructured RNAs is also true for the E. coli version. So that sort of just makes sense. It's a general thing, but we don't know what gives it the specific nucleotide preference. So we sort of, you can easily see this homology between the two proteins, but we don't know to that level of detail how it is that they're interacting with those 5' ends that gives the nucleotide specificity. So we can't really say what it is that's different about it that's causing the difference. Oh, that's a good question. We haven't tested that. I don't know. I think based on the structure of the E. coli RPPH, it only interacts with a few nucleotides. So probably not very much. Probably RPPH could come in very early on. Yeah. Any other questions? Thanks, everybody.